All right, that gets us through our vascular tissue, synapomorphy, and gets us through this vascular tissue one. And it moves us on to looking at the evolution of leaves. And if you'll notice on here, we actually have two different types of leaves that I put on here. Lycophytes or lycopods have what we call microphyllous leaves, where the rest of vascular plants have what we call macrophyllous or megaphyllous leaves. Those mean the same things. Megaphyll is what you'll see these referred to most of the time. And so we have these two, so vascular plants go along with vascular plants is the evolution of leaves and roots. So these are, but these evolved independently, and leaves evolved independently in these two major groups. So the first group of those is the lycophytes, as I mentioned before, lycophytes. And lycophytes have microphylls. And although microphylls are small, we have some megaphylls that are small too. But microphylls, typically very small, and they have one central vein in the middle of them, and that's it. And I'll show you why that is. Where in our ferns and seed plants, so ferns and seed plants, we have what we call megaphylls, so megaphylous leaves. Megaphylls, as we'll see, are typically larger, and they have branched veins throughout them. And that helps us distinguish those morphologically from each other. Microphylls with their single vein, and megaphylls with their multiple or branched veins. And so what I want to do is remind you real quickly about these first polysporangiophytes and these first polysporangiophytes and their branched sporophyte structures. And so those, and I told you that these got bigger and then acquire vascular tissue sort of through time. Well, those first vascular plants then probably looked a lot like these, where they had just this sort of dichotomously branching axis, and that dichotomously branching axis didn't really have leaves associated with it, and maybe it was probably photosynthetic, similar to a hornwort sporophyte, but didn't have much surface area for photosynthesis. This goes into the evolution of megaphylous leaves from these dichotomously branched axes. And so this is something that's called the telome theory for the evolution of leaves, or specifically megaphylous leaves. And so just to make sure that I have this written down for you, megaphylous leaves have multiple and or branched veins in them associated with them. And so this telome theory is actually pretty straightforward. And there's fossil evidence throughout those polysporangiophytes that kind of show this progression that I'm going through right now. So there's 3 steps that happen and that we are hypothesized happened in the evolution of the leaves. The first is that these started from these dichotomously branched axes. Dichotomously branched vascular axis. So these are dichotomously branched sporophytes with vascular tissue inside of them. The first step to that is differentiation of these into a trunk and lateral branches of the axis into a trunk with lateral branches. And so here we have the trunk part with these lateral branches, and these, rather than being sort of evenly dichotomously branched, they've got varying varying sides to that, and we see that this actually proceeds into then taking those lateral branches and planation of the lateral branches. Planation of the lateral branches. And so what do I mean by planation? So in the dichotomously branched thing, if you think of this in 3D, these are evenly dichotomously branched all around. And then as we go in, we differentiate into a trunk with lateral branches. These are, again, 3D, branching out on all sides. When we have planation, we flatten that out so that that branching is happening in one plane. That sets us up for the third step in the coelom theory for the evolution of leaves, and that is webbing of these lateral branches. And so if we have that photosynthetic tissue then fill in between those lateral branches and make webs, well, what do we have in the end? We have a complex leaf, or a megaphyllous leaf, a leaf that has multiple branched veins inside of it and flattened out, planed out webbing. That's all that photosynthetic tissue or the leaf surface in there. Well, that's our theory for the evolution of leaves. And then and as I mentioned, we actually see these intermediate steps in the fossil record, which is really cool. Okay, so looking back at our phylogeny here, we'll see that these leaves evolved multiple times independently. Once in lycophytes, where we have our microphyls. So those microphyls just have one single vein in it, a single vein down the middle of it. And then our megaphylls evolving along the lineage that leads to ferns and to seed plants. Okay, so let's take a look at some of the let's look at some of the diversity of some of these and the phylogeny, just so we can see. This phylogeny is a little different than we're used to looking at it. And I'll just point out this is the phylogeny that includes bryophytes down here at the bottom. So there's our here's our paraphyletic bryophytes. There's our hornworts, and then these are some mosses and some liverwort groups. So there's our bryophytes. And so this would actually be vascular plants is what this node would be right here, is our vascular plants. And if we look at those vascular plants, we've got a couple of different lineages. The first of those that I want to highlight is the lycophytes. And so this lycophyte includes this group that we call the club mosses. The rest of this includes our seed plants, where most of the diversity is. And then our other clade here is the fern clade. And I want to focus on the lycophytes to start with. If we look at, I've put on the side of this slide the diversity, we see that in lycophytes, there's only about 1,000 species or so of lycophytes. And so here's some pictures of lycophytes. Lycophytes are also called ground pines. This picture right here in the middle is one from Michigan, actually, but this occurs throughout temperate regions. And we see this small, these are typically about 4, 5, 6 inches tall. They look like a little pine tree almost on there, often common name called ground pines or club mosses. But I also include some tropical ones like the Superzia here. And then a couple of other genera, Selaginella and Isoedes. And Isoedes is one that grows also around here. It's quite diverse in both the temperate and tropical regions. And I wanted to remind you that during the Carboniferous, the lycophytes are actually one of the dominant one of the dominant things in one of the dominant land plant groups, and actually grew huge. So while most of these are small right now, these used to be tree-like, up to 40 meters tall. All those modern coal deposits are those. And so here's some artistic renderings from fossils that are found throughout the world, showing these very tree-like or tree-formed arborescent lycopods. Super cool. I wish I could have seen these growing. These are awesome. Let's look at our lycopodium here, as an example of that. This is one that grows. This is one, like I said, that grows around here in temperate regions. This is that ground pine, and we see the microphyllous leaves. And so here's here's one of those leaves associated along these axes. These are going to have single veins associated with them. They also have a homosporous life cycle, and I'm emphasizing this because well, you'll see in just a second, in other groups of lycophytes, they've actually evolved another characteristic. But what homosporous means is they have one type of spore. And so when we look at the life cycle of these, we see our standard land plant alternation of generation life cycle. And we have this one spore type, as I mentioned. Oops, one spore type. And so we have meiosis producing the spores. That spore then grows into our gametophyte. The gametophytes in these are free-living gametophytes, like we see in ferns. They're free-living, small. Typically, these are just under the surface of the soil, just small, and they look like a little clump of dirt. But they produce archegonia and antheridia, just like in ferns. And they are, because we have just this one type of spore, these are also hermaphroditic. And so they produce both male and female reproductive structures on the same free-living gametophyte. I'm not going to go through the rest of this life cycle because we've gone through several of these, but we end up with our standard alternation of generations life cycle, keying here on this homospory, just having one type of spore associated with them. One of the other groups that we see in lycophytes is the genus Selaginella. Selaginella is about 700 species or so. The main reason I want to bring these up is that they are heterosporous. And what that means is that we have two types of spore. A cool plant that you may or may not be familiar with is the resurrection plant. And this one is shown here. These are found in the desert southwest of the US. And in the dry season, these will actually dry up into this sort of crusty brown form. You might walk by this little ball and think it's a dead plant right there. But if you get these wet, as soon as they're hydrated, they actually open up, turn green again, and begin to photosynthesize. And so they live in moist habitats, but they can survive in drier places because they have this ability to resurrect. We have some of these in lab that I would have shown you that we dry up every year, and then our simulated monsoon season is that lab section that would have happened next week, and we put these in a pot of water. And these actually uncurl, open up, and turn green and start to photosynthesize again. Very cool. But not only this species that we call resurrection plant does this, actually we get this in other Selaginella as well. All right, so let's look at that heterosporous life cycle real quickly. When we look at heterospory, the main point that I want to make here is that we have two types of spores now, not just one type of spore. And we have what we call megaspores, which are female, and microspores, which are the male. These are called mega and micro because of their size. Megaspores are much larger than the microspores. Typically, there's fewer number of megaspores as well, so you put a lot of investment in the female spore and little investment in the male spore, and I think that's often a common theme is to have lots of investment in the female reproductive strategy and make males small and cheap, and I think that translates well to our society as well. And that's a little joke. So once these germinate, then instead of having hermaphroditic gametophytes, these actually have separate megagametophyte, so a female gametophyte, and microgametophyte. The megagametophytes will go on to produce the eggs, and the microgametophytes will go on to produce the sperm. And then fertilization occurs and we go through the rest of the life cycle. So we've got two different types of spores there. And this is a really important variation that we haven't seen before in the life cycle, and that's that heterospory. So here's looking at sporangia in Homosporous Lycopodium, and looking at sporangia in a Heterosporous Selaginella. And so sporangia are where the spores are made, and we can see these large megaspores, few large megaspores, and then we can see a microsporangium with lots of little microspores in there. Heterospory, I emphasize this here because heterospory actually is, we see heterospory evolve again we see this evolve again in seed plants. And so we see this in both the lycophytes and in some of the lycophytes and in the seed plants. And so there are a couple different things that are about heterospory that I want to mention as well. One is that in heterosporous plants, so this is in both in anything that's heterosporous, the gametophytes are endosporic. And so these gametophytes are reduced even more. So the gametophytes develop inside of the spore wall. So remember, in Lycopodium and in ferns, that the spore goes out and germinates into this small but free-living little gametophyte, what makes Antheridia and Archegonia and sperm and eggs, and then fertilization, and we go to the sporophyte. In Heterosporous Selaginella and Isoetes, and then in seed plants, the gametophyte actually is reduced that much more and develops inside of the spore wall. And so here we have our little tiny microspores, and inside that, the male gametophyte develops. And so it's reduced the Antheridia are actually inside of that spore wall, where sperm are then produced. The megaspores is where the Archegonia are produced inside of those megaspores, so inside the spore wall. So no longer are we germinating and growing into a separate plant, but we're actually doing this all inside of the spore wall. Everything in the gametophyte is now reduced quite a bit more. And so here in Selaginella, we can see the megaspore wall. That this one would have had an archegonium and an egg inside of it. That egg gets fertilized by sperm. Those sperm are swimming sperm again still. And we have the young sporophyte coming right out of that spore wall. Okay, so that's our heterospory. I want to just take the last few minutes here and go through briefly the most diverse group here which is our fern groups. And so let's see. I lost my phylogeny. I'm going to scroll back up to my phylogeny real quick. There's my phylogeny of the vascular plants. We just went through our lycophytes, and I want to go real quickly through the ferns themselves. And ferns are made up of a couple different groups. I'm not going to spend a ton of time on these, but I want to just talk our way through some of these some of that fern diversity for you. And so most of our ferns are what we call leptosporangiate ferns. These are what you're most familiar with. These are ferns with large megaphyllous leaves, fronds, as we often call them, that unfold from a fiddlehead. So these like uncurl. That's called cercinate vernation. Really cool words. And have very thin-walled sporangia, and that's what leptosporangiate means, is thin-walled. So the sporangia themselves are very thin. Those sporangia, which here's an individual thin-walled sporangia shown, are arranged on the bottom of the leaf into groups called sori. So I think many of you have looked at the bottom of a fern leaf before and seen a bunch of little dots on there. When you look, if you were to look at one of those little dots with the microscope, this is what you would see over here. So this is a section through a sorus, and what that sorus is a group of a bunch of little sporangia. Each one of those sporangia contains within it a bunch of spores. So these are those thin-walled sporangia that we see in our leptosporangiate ferns. This is just a life cycle of that fern shown here in a little bit of pictorial form. And so here we have our sporophyte on the left. On the underside of that, we'd have those groups of sporangia. Here's one individual sporangia, and those thin-walled sporangia actually open up and then catapult the spores out. And those spores then fly through the air and will land somewhere and germinate and develop into their separate free-living little photosynthetic gametophyte. That gametophyte makes both antheridia and archegonia, and the sperm then have to swim and find the archegonium, swim down, fertilize the egg, and then that spore can go, fertilize the egg, and then that can develop into, again, this mature sporophyte. So leptosporangiate ferns, or these thin-walled ferns, are some of our most diverse ferns. They range from tree ferns, like I said before, temperate ferns like our maidenhair fern, and many different, over 11,000 species, and a lot of tropical species as well, and some that don't look quite like ferns like we're used to seeing. And in lab, we would have seen some of this fern diversity. All right, the other group of ferns are called eusporangiate ferns, and eusporangiate ferns are are thick-walled sporangia. And so these actually have multiple cell layers thick to their sporangia. These are larger, they're easier for people to see, and they're not typically grouped into all of those sori like we saw, the sorus like we saw before. And so here's some of those thick-walled sporangia. So we see them arranged on the underside of the leaf, but each one of these is an individual sporangium on there. This is a plant called adder's tongue, and we can see the sporangia arranged along this fertile part of the leaf here. And then this is called a moonwort, and moonworts also have these thick-walled sporangia, sometimes called grape ferns, because the sporangia are large, thick-walled sporangia. Okay, The last two groups of the eusporangiate ferns are the horsetails and the whisk ferns. And both of these are things that were previously not considered to be ferns. But molecular phylogenetic evidence has shown pretty clearly that both the horsetails and the whisk ferns are part of this fern clade, solidly part of this fern clade. And they're actually most closely related to other eusporangiate ferns. And so now we consider these to be part of the eusporangiate fern groups. They have a lot of morphologies that are unique to themselves that other groups don't have, which is why they were classified separately to previously. But now they're actually shown, like I said, to be part of that group. And we can look at them, and what we see is they're odd. They have eusporangia in that they have these thick-walled sporangia. This is the whisk fern, Xylodon. So they've got these thick-walled sporangia, but their leaves are very small and reduced. And so the leaves were previously interpreted to be microphyls because they're so small and lack branched vascular tissue. But what we've now seen is that these are actually just reduced megaphils and specialized for the morphology of this particular individual. And the same thing is true of the horsetails. These are our horsetails. Or Equisetum is the genus of these. And these also have these very reduced leaves. And so, again, these were considered to be a separate lineage. They thought they were more closely related to lycopods, and they're actually not. They're part of the fern clade. And so we've now reinterpreted these as just having very reduced megaphils and more closely related to other eusporangiate ferns than they are to lycopods. I'll just add here that along with those tree-like lycopods in the Carboniferous, we also had tree-like horsetails, so up to 20 meters tall. This is one called Calamites. These are well known in the fossil record, but are and morphologically are really similar to what you see today in extant horsetails, but are actually 20 meters tall. This is another one that I really wish I would have seen. These amazing arborescent ferns that were arborescent horsetails. Okay, I'm going to stop here and just with the sort of preview that we're going to move from here now into seed plants and talk about our synapomorphies that we find and innovations that we see in seed plants. I know you're excited. Stay tuned.